Good afternoon, everyone. It's my pleasure to welcome you to the 2017 Warren K. Lewis Lecture in Chemical Engineering at MIT. Before I introduce our eminent speaker, I'd like to tell you a little bit about the named series and Warren K. Lewis. Warren K. Lewis is actually one of the fathers of our department, and he was affectionately known as Doc Lewis by his students and associates. He had an enormous impact on shaping this program and the field of chemical engineering as a whole. Doc Lewis received his PhD in physical chemistry from the University of Breslau, Germany in 1908. He then joined MIT as an assistant professor in 1910 and was promoted to professor in 1914. He became the first head of the newly formed Department of Chemical Engineering. Chemical engineering, before that, was a program in the Department of Chemistry. When it became a full department, Doc Lewis, who led the charge, was able to fully separate from chemistry between 1920 to 1929. And after this, he devoted himself to teaching and research, as well as consulting. He remained an influential member of the department until his death in 1975 at the age of 92. Now, Doc Lewis actually revolutionized the design of chemical engineering equipment with the concept of unit operations, the first paradigm of chemical engineering. He was an eminent researcher and inventor. He contributed much to the field of industrial chemistry, with over 80 patents to his credit. For example, he pioneered the use of fluidized bed catalytic cracking and refining. Above all, Doc Lewis was a superb educator, and here you can see a picture of Doc Lewis right up here at the front pontificating with his students. His text, Principles of Chemical Engineering, with Walker and McAdams in 1903, was first defined the discipline. His lectures are legendary for the combination of beautifully organized material and Socratic exchanges with his students, as you can see. Not surprisingly, Doc Lewis won many awards, among them the President's Medal of Science, the Presidential Medal of Merit, and the Priestley Medal from ACS. As a most fitting honor, our professional society, the American Institute of Chemical Engineers, has honored him with the creation of the Warren K. Lewis Award, which recognizes outstanding education in chemical engineering. The Warren K. Lewis Lectureship here at MIT was established in 1978 to recognize Professor Lewis's revolutionary impact on chemical engineering. By developing the concept of unit operations, he revolutionized the design of chemical engineering processes and equipment. Throughout his career, Professor Lewis was mindful of the needs of industrial practice. Accordingly, the Lewis Lecture features speakers from industry and academia in alternation. Our speaker for today, David Terrell, is the Ross McCullum William H. Cochran Professor of Chemistry and Chemical Engineering, Provost and holder of the Carly and Shirley Larson Provostio Chair at California Institute of Technology. Dave was educated here at MIT and then the University of Massachusetts at Amherst, where he earned his PhD for research done under the supervision of Otto Vogel, who is one of the recognized fathers of polymer chemistry. After a brief stay with Takeo Sakusa at Kyoto University, he accepted his first assistant professorship in the Department of Chemistry at Carnegie Mellon in the fall of 1978. Terrell returned to Amherst in 1984 and served as the director of the Materials Research Laboratory at UMass before moving to Caltech in 1998. There he served as the chair of the Division of Chemistry and Chemical Engineering at Caltech from 1999 until 2009. Now, Dave's research interests lie in macromolecular chemistry and in the use of non-canonical amino acids to engineer and probe protein behavior. His contributions to these fields have been recognized by a large number of awards and recognitions, among them his election to the American Academy of Arts and Sciences and to all three branches, Sciences, Engineering, and Medicine of the U.S. National Academies. He's one of the rare members who is in all three of the academies. As you'll see from his talk today, David has always been at the frontiers of bioengineering. He introduced new concepts to the use of precision biology and cell engineering as a route to novel polymeric materials, the use of protein engineering as a means of implementing non-native or non-canonical amino acids into biomolecular backbones. Dave has done a great many things to further the fields of chemistry, biology at the interface with chemical engineering, and it's thus contributed to the expansion of our field in these areas. But Dave is also one of our alumni. He was a very active student when he was here at MIT, as you can see, class of '74. He was active in part because of his research, but also because of his activities. And this is one of them. He's an avid baseball player. And this is the MIT baseball team, varsity, 1973. And Dave, if you can't pick him out, I've given you a chance to guess, is number 3, right here. There he is with the, looks very cool, and I like the hair, too. It's very, very engaged. Now, it turns out that he is, and always was, a natural leader. So the next year, he became captain of the baseball team. As you can see, he also developed a much better eye protection that year. But along with these things, Dave was a UROP for Professor Ed Merrill, who was a pioneer in the field of biomedical engineering and polymers for biomedical applications. Here we can see Dave at our celebration, in fact, of Ed Merrill's symposium. And actually, a quote from Ed that was given to me very recently is that Dave is my most illustrious example of a UROP student. And so Dave is the epitome of that. Here we can see him with some of the other alum from the Merrill Lab. Dave also gave our 2007 Alan S. Michaels Lecture. He has helped the department in many ways, both visible and behind the scenes, by serving on our visiting committee for 2 terms, 2009 and 2011, 2013 and 2015. He serves on the chemistry visiting committee now from 2015. He's written infinite numbers of letters, helped to support numerous of our faculty members, but most of all, he's impacted us in our field in important ways. You can see, in fact, that he has, this is just a couple of offspring from his lab that are current faculty, but there are more. I just couldn't manage it on one slide. He has served as a true leader in the field of chemical engineering. So I believe he is very worthy of this Warren K. Lewis Lecture, and we're very excited to hear what he has to say. Thank you, and let's welcome David.
Thank you, Paula, for most of that introduction. There were some parts that were really good in that introduction, and I appreciate it. I certainly appreciate the obvious effort that went into digging out that history. That history is virtually untraceable, and so the fact that Paula was able to find that information should worry all of you if you think you've been able to hide anything from your department chair. You have not. So let me thank you again for a wonderful invitation. It really is a terrific honor to be part of this lecture series, in part because of the association with Professor Lewis, who, as Paula said, has been an enormously important figure in the field and at the institution, so it's wonderful to be associated with him, and then also with the previous speakers, a wonderful group of speakers. So I appreciate all of that. I'm a little concerned that the slides are not showing. They should be, I think. Thank you. I wish everything were that easy. Thank you very much. And so I'm going to talk to you for the next 45 minutes or so about this subject of reinterpreting the genetic code. In keeping with the historical flavor of Paula's introduction, I'm going to start with a little bit of history. I'll talk a little bit about how that history shaped the decision to enter this field at the intersection of materials science and biology. And then for the second 2/3 of the talk or so, maybe even more than that, I will tell you about a recent project, in fact, an ongoing project, where we're using this strategy of reinterpreting the code to engineer therapeutic proteins, in particular insulin for treatment of type 1 diabetes. I've used this title on previous occasions, but it seems especially timely today because, as you may know, this is the 50th anniversary of the award of the Nobel Prize to Robert Hawley, Gobind Khorana, one of your longtime colleagues here at MIT, and Marshall Nierenberg, and the citation is for their interpretation of the genetic code and its function in protein synthesis. And as I'm sure most of you know, what the genetic code does is to tell us how the language of nucleic acids can be translated into the language of proteins, a sequence of nucleotides in RNA read out by the cell in terms of a sequence of amino acids in the protein. And for roughly the last 25 years, we've been asking the question posed at the bottom of the slide, what if we could change how the cell interprets the genetic code so that there could be a single RNA message made in the cell, but then it would be read out in the form of different proteins. Those proteins might be related to natural ones, or they might be wholly artificial. We came to this field through an interest in macromolecular chemistry. And as Paula said, my interest in macromolecular chemistry was stimulated by Ed Merrill. This is my favorite picture of Ed because it looks like he is about to take flight, something that I thought was going to happen frequently in the course of the lectures that he was giving in 1972, 1973. I took 2 courses from Ed. I did at least 2 years of research in his laboratory. He brought Paul Remp to MIT during that time. He brought Paul Flory to MIT. I took 2 courses from Paul Flory. All of those experiences were enormously important to me, and I remain grateful to Ed for what he did those 45 years ago. He's a wonderful person, wonderful teacher, inspirational mentor for many, many people. So as all of you would know, long-chain molecules are of great importance, both in technology and in biology. I'd ask you to take a quick look around the room and estimate how much of what you see is made up of long-chain molecules. The answer is essentially everything. The exception to that might be the blackboards. The concrete looks like it's not polymeric in nature, but in fact it's covered by a polymeric coating, so in fact what you're seeing there is almost surely macromolecular. And so the special properties of chain molecules allow them to do biological and technological jobs that no other class of molecules can do. That shows up in biology in the form of enzymes. This is one that you've certainly heard about, cyclooxygenase 2. This was very much in the news during the Vioxx concerns. This is a target of Vioxx, non-steroidal anti-inflammatory. This is Herceptin, an antibody that's used for the treatment of breast cancer, and of course then the iconic double helix of DNA. In synthetic macromolecules, we're familiar with different kinds of behavior, or at least we take advantage of different kinds of behavior. Polyethylene, a tough, flexible material, polymethyl methacrylate, optically clear and stiff. And so one of the things that becomes apparent from looking at this combination of macromolecular systems is just how strikingly different the functions are for biological and synthetic polymers, despite the fact that they share long-chain character. Now that comes from the different ways in which they're made. Polymers in technology are typically made through processes that could be roughly represented as shown at the top of the slide. Generation of some sort of a reactive intermediate, maybe a free radical, adds to an olefin. That process continues. That's a process that can be controlled in an average sense. We can control how long the chains are on average, but we always have competing initiation, propagation, and termination steps, and so those chains are always complex molecular mixtures. And so while the well-defined molecules of biology do molecular recognition, they do catalysis, they carry information, they transmit information from generation to generation, the complex molecular mixtures of synthetic polymer chemistry are largely used as structural materials. They have wonderful structural properties, so I'm not trying to make the case that somehow the uniformity of biological macromolecules makes them better, but in fact it is very difficult to encode information into the complex molecular mixtures that are characteristic of synthetic polymers. Biology avoids that, of course, by using a template to make polymer chains. In the case of proteins, it's a messenger RNA template, and we're looking at the genetic code here. The genetic code is actually the interaction between the nucleotides in the messenger RNA and the nucleotides in a transfer RNA that then delivers a an amino acid to the end of the growing polypeptide chain so that the length and sequence of the protein chain are completely defined. So as you can imagine, if you need the informational properties that allow you to do catalysis, recognition, and information transfer, you need this kind of control at the level of sequence and length. Now, no matter how complicated you think synthetic polymers are, I bet they're more complicated than you think. And I'm going to show you an example of that. Here's a polymer that we've already talked about. This is polymethyl methacrylate, lucite or plexiglass. This is the repeating unit structure. When you make this polymer by means similar to what I showed on the previous slide and you put it in a mass spectrometer, you see all the different chain length variants that we always get in chemical polymerization processes suggesting heterogeneity. But let's imagine that we could control the chain length absolutely, and we could make a 500 mer, which is roughly what the commercial polymer is. How many different structures would there be? There would be 2 to the 500th different structures, because the stereochemistry at this position is essentially unrelated to the stereochemistry at the subsequent position. And so for a 500 mer, you've got 2 to the 500th possibilities. Now at MIT, you don't have to explain what 2 to the 500th is. In other audiences, I've done that, but I just like all those zeros. It gives you, I think, a more visceral sense of just how big a number this is, and what are the consequences of the size of that number. It means that although we've been making polymethyl methacrylate for more than 50 years on an industrial scale, we've never made the same molecule twice. There are just not enough molecules to populate all of those stereochemical possibilities. And so this really distinguishes the two fields of macromolecular chemistry. And again, I want to come back to the point that this kind of heterogeneity can be an advantage. We use polymethyl methacrylate because it's optically clear. It's optically clear because it doesn't crystallize. And it doesn't crystallize because it's stereoirregular. And so there are reasons why we would like molecular systems to be heterogeneous, but it does determine what they can and can't do. It also means that there's been kind of a troubled relationship between the sciences of natural and synthetic polymers over the years. Most people would identify the founder of polymer chemistry as Hermann Staudinger. Paula didn't go back quite far enough to detect the fact that Hermann Staudinger is my academic great-great-grandfather. And I'm very proud of that fact. We never met one another. He gave his Nobel Prize lecture in 1953, the year I was born, so our direct contact was very limited. But here's Staudinger, the founder of polymer chemistry, and what he talks about in his Nobel lecture is the masterly macromolecular architecture of living matter. And this reflects the fact that in the early days, people didn't distinguish between these two fields. Staudinger was just as interested in polystyrene as he was in cellulose. And in fact, when he made polyoxymethylene, he made it as a model for cellulose. And so he was very much trying to understand natural polymers when he made synthetic ones. And so if you go back to the origins, I think most people would cite this paper in 1922 as the confirmation of the macromolecular hypothesis. One of the things that I learned about from Ed Merrill was the controversy in the 1920s about whether macromolecules were covalently linked long chains or whether they were non-covalently associated colloidal systems of one kind or another. It was known that natural rubber was chemically unsaturated. It has double bonds. The hydrogenation was believed to change the intermolecular interactions, the non-covalent interactions. And so what Staudinger found was that hydrogenation didn't change the apparent molecular weight. And the absence of that change, under conditions where the intermolecular interactions would have to have changed, was regarded as evidence for the covalent chain-like structure of macromolecules. Now, as I was doing my digging into the history in preparation for this lecture, I came across this paper by Warren Lewis and McAdams, and I would point out that it's from the Department of Chemistry and Chemical Engineering at MIT, and the date is July 1920. So this was before the Staudinger and Fritchie paper, and what Lewis and McAdams were doing was trying to develop analytical methods that would allow them to determine the hydrocarbon content of natural rubber preparations. And what they decided to do was to brominate those double bonds, very much in the spirit of what Staudinger was doing. And then is this kind of caveat down here: inasmuch as this paper will not deal with the structural formula of rubber, the conventional formula, C10H16N, will be used to designate rubber hydrocarbon. He wasn't ready to commit to the macromolecular hypothesis. That wasn't really his primary interest. He just wanted to know how much hydrocarbon was in there. Around that same time, there was a second set of experiments that's generally regarded as an important confirmation of the covalent structure of macromolecules, and that was the independent measurement of the molecular weight of hemoglobin by osmotic and ultracentrifugation methods. And the fact that these two independent methods gave the same result was also taken as evidence of a covalent structure. And again, I would emphasize that hemoglobin was very much cited by the polymer chemists at the time as evidence for this macromolecular chemistry. People were not distinguishing between natural and synthetic polymers in any fundamental way. Now we get to the amateur history of science. This is my own interpretation of what happened, and I'd be glad to discuss it with anybody who would like to do that, whether you agree or disagree. I think what happened in terms of the way the field developed is that after this period of time, the 30s through the 60s, the two fields really did diverge. And the reason was that each field had its own set of problems that were really compelling. And there was no need during this period of time for the two fields to look across the boundary at one another. It was discovered that synthetic polymers had their remarkable physical properties. They could be processed easily, they didn't cost much, and this really created the soft materials revolution of the 20th century. At the same time, the biochemists and biophysicists began to understand the structure of DNA and proteins and the connection between those structures and the biological functions that we talked about a few minutes ago. So through the midst of the 20th century, and in fact around until the time that I began to think about polymers with Ed Merrill and others, these were really two different fields. The two disciplines really didn't talk to one another very much. They began to converge a little bit later, at least conceptually. I'm not sure this was widely perceived at the time, but on the synthetic side, there were advances in synthetic methods that showed us what the value would be of improved architectural control. Ziegler nanopolymerization gave us linear polyethylene and isotactic polypropylene, resulted in the Nobel Prize for Ziegler and Nana in 1962. Living polymerization, developed by Michael Schwartz in 1956, gave us a kind of rudimentary control of sequence. For the first time, we could make block copolymers and telechylic polymers with specific end group functionality. And the utility of that architectural control began to become apparent. At the same time, biology began to emerge as an engineering discipline. The key paper here, of course, was the 1972 paper from Paul Berg's laboratory introducing recombinant DNA technology. There were then advances in DNA synthesis and sequencing led by Khorana, Letzinger, Sanger in terms of sequencing technology. And so by the mid-1980s, we had reached a point where DNA could be made, sequenced, cloned, and amplified even by a polymer chemist. These methods had become routine, they'd become transferable, and it was at that point that we decided that we might be able to use recombinant DNA technology as a new way to do synthetic polymer chemistry. And so we proposed and have been working for many years now on a scheme of this kind, where we design primary sequences of amino acids that we think will behave in interesting ways. We take our designs in part from structural biology, in part from polymer chemistry and physics. We encode those amino acid sequences into complementary DNAs. That's where we use the genetic code. We then make the DNAs, or just order them now in full-length form from the biochemical suppliers. And then in order to get from the coding sequence to the molecule that we really care about, we use recombinant strategies, meaning that we put the coding sequence into a plasmid, a circular DNA. We reintroduce that recombinant plasmid into typically bacterial cells, but it doesn't have to be bacteria. And then we grow those cells in a flask or fermenter under conditions where they'll make the polymer for us. And this really changed the way we could think about macromolecular chemistry, because for the first time it gave us essentially absolute control of the molecular architecture, the length, sequence, and stereochemistry of the chains. So here's an example of polyglutamic acid, which can be made either by chemical polymerization, that is the ring opening polymerization of the leucine hydride, or by gene expression. If you do it by ring opening polymerization, you see a mass spectrum that looks just like the polymethyl methacrylate mass spectrum. On the other hand, if you make an artificial gene that encodes polyglutamic acid, you express it, you put that product into a mass spectrometer, you see a single molecular ion, indicating that the molecular architecture is well defined. If you want the chains to be longer, you just make the gene longer. There are some other things that come along with this. There's access to well-defined folded structures, so you can start to control molecular assembly and the formation of macroscopic materials. One can incorporate functional sequences derived from natural proteins, building biological information into materials. Can amplify and evolve things if you don't get them quite right in the first place, or maybe you don't know what you want to make in the first place. You make a library, sort through it, iterate on that process, and find what you want. And then something that we didn't anticipate at the outset, and I talked a little bit about yesterday and will again today, is that this way of thinking about things has led us to some new approaches to both clinical and fundamental biological problems. That's the good news. For a polymer chemist, there's bad news, and that is that nature uses essentially 20 amino acids to build proteins, and that's enough to do catalysis and molecular recognition to support all known forms of life, but it's not enough to do interesting polymer chemistry. And so one of the things that has occupied us for the last 25 years is how to expand this palette of amino acid building blocks as cells make proteins. So this is the notion of reinterpreting the genetic code as we go from DNA to RNA to protein. And the conventional view, of course, is that once the messenger RNA has been made in the cell, the genetic code tells us what the sequence of the protein has to be. And we saw, those of you who attended the lecture yesterday saw that this doesn't have to be true. It's relatively straightforward to reassign a codon from one amino acid to another, and in that way, to effect what we call alternative translations. That is, a single message can lead to many different proteins depending on the interpretation of the code that's operative in the cell at the time that protein is made. So how do we reassign codons from one amino acid to another? The key step here is the step shown over on the right-hand side. This is the process by which an amino acid gets into protein. We showed some of this process a few slides ago, but there's an early step here that is absolutely essential. That's the activation of an amino acid by an aminoacyl tRNA synthetase, which then attaches that amino acid to the 3' end of a transfer RNA in the aminoacylation step. That's the most stringent step in the process. If you can get a new amino acid onto a transfer RNA, it will almost surely go into protein. There are a few exceptions to that, but not very many. And if you think about that a little bit, it's not terribly surprising. The ribosome, for example, has to be able to accommodate all 20 amino acids. So it can't examine the amino acid side chains terribly carefully, but the synthetases are adapted individually to each amino acid, and they examine the side chains very carefully. And so if you can modify the synthetases of the cell, or if you can just find amino acids that are good enough substrates for the wild-type synthetases that they can be put onto these transfer RNAs, they will be read out according to the anticodon in this aminoacyl tRNA. That's the process of reinterpreting the genetic code or codon reassignment. And it can be done either for sense codons or for nonsense codons. So we spent the last 25 years inventing new amino acids and synthetases to activate them. And by doing that, we've been able to introduce 50 or 60 new amino acids that cells can use to make proteins. This is nowhere near a complete set, even of the amino acids that have been developed in our laboratory. And there are many laboratories around the world that do this, that have done nicely complementary things. And so if one were to show a complete set, you wouldn't be able to see any of the structures. You would just see that there are lots of structures up there. That doesn't seem like such a useful presentation to me. And so the amino acids here are arranged as analogs of the natural ones. That means that everything along the top can be regarded as a methionine analog. It will be activated by the methionine tRNA synthetase and put into proteins in response to methionine codons. The color code shows what you have to do. For the black ones, you don't have to do anything at all. You just squirt them in. They're good enough substrates for the wild-type synthetases that they'll be put in competition with methionine. And so if you want to label proteins lightly with azide side chains, which is what we were talking about yesterday, you literally just squirt AHA, azidohomoalanine, into the cell. But if you would like to read out all of the methionine codons as AHA, you use a methionine auxotroph. That means a cell that can't make its own methionine. You then deplete methionine from the medium and add AHA instead. And when you analyze proteins made under those conditions, you typically won't find any methionine. All of the methionine codons will have been read out as AHA. The ones shown in blue are also activated by wild-type synthetases, but very slowly, so you have to overexpress the wild-type synthetase. And the red ones and the green ones require mutant synthetases. The red ones require active site mutants, the green ones require editing site mutants. And so I think most people working in the field, and probably even people looking in from the outside at this field of non-canonical amino acids, would agree that, with few exceptions, if you want to get some reasonable amino acid into a protein made in cells, you're going to be able to do it. This chemistry turns out to be a lot easier than I thought it was going to be. And then you come up against the only question that my mother ever asked me about my research. So what? So what, David? I didn't care that there were 20 amino acids. Why should I care that you now have 60 new ones? What is this good for? And I think that that is clearly an important question that we should all be asking ourselves about our research programs. Why should anybody care about this development? And so what I'd like to do is to show you just one example from our recent and, in fact, ongoing work where we've used the non-canonical amino acids chemistry to re-engineer insulin for the treatment of type 1 diabetes. I think everybody probably recognizes the extent of insulin as the extent of the problem of diabetes worldwide. Roughly 400 million people suffer from diabetes, about 30 million in the United States, about 9% of our population. There are two types. Patients don't produce insulin in type 1 diabetes. In type 2, they're insulin resistant. They don't respond to insulin. Both forms of diabetes are treated by subcutaneous injections of insulin. And insulin is one of the genuine miracles of 20th century medicine. There's a wonderful book about the discovery of insulin. That's actually its title, The Discovery of Insulin. It's written by Michael Bliss, and it tells the heroic story of the original purification of insulin in the 1920s. In fact, right at the time that the macromolecular structure of polymers was being established. I would recommend the book. It's one of the best history of science medicine books I've ever read. So insulin allowed a miraculous change in the quality of life for people, especially with type 1 diabetes. With type 1 diabetes, it's often called juvenile diabetes, a disease of young people. Lifespan was short, quality of life was not good. Once insulin was available, those same people now live full lives with high quality of life. Nevertheless, insulin is not ideal as a therapeutic. It has at least two problems. It acts more slowly than we would like after injection, and it's prone to fibrillation. It's unstable with respect to amyloid-like fibrils. Some of you may use insulin, or you may have friends or family members who do. And if that's the case, you've almost surely seen evidence for this first point, that insulin acts too slowly after injection. When a diabetic patient is going to eat, he or she will take a blood glucose reading prior to the meal, estimate the needed dose of insulin, and then inject. The reason for that is it takes too long for the insulin to get from the injection site into the blood. If you were to wait for the blood glucose to begin to rise, you wouldn't be able to manage the blood glucose properly. And so, if we're ever going to have self-regulated insulin delivery systems that respond to a rise in blood glucose, we'll need to have insulins that act faster. Insulin fibrillation is apparent to anybody who uses an insulin pump. Typically, you'll get fibril formation in a pump after something like 3 or 4 days, and so the insulin has to be recharged into the pump. If we're going to have self-regulated systems, we can't be recharging them every few days. And so what we did was to set out to see if we could address these two problems. They have a common molecular origin. And that common molecular origin is the fact that the monomeric form of insulin, which is the active form that binds to the receptor, is only marginally stable. Nature solves that problem by forming a hexamer in the pancreas. The hexamer is the stable form, and that allows insulin granules to be stored in the pancreas for prolonged periods of time. The pharmaceutical companies also formulate insulin as a hexamer. That's the structure of the insulin hexamer up here. It's a zinc protein. There are 2 zinc ions in the hexameric structure. And the dissociation of that hexamer is the rate-determining step in getting insulin from the injection site into the blood. So you might ask, why not just use insulin in its monomeric form? I mentioned that it was unstable. It's unstable, believed to be unstable, because in the monomeric form, the chain ends begin to fray, and those frayed, flexible chain ends begin to aggregate, form nuclei, and eventually grow into long fibrils. There's a good strategy for making faster-acting insulins, and there are fast-acting insulins on the market. And in fact, the pre-meal routine that I described almost always involves a fast-acting insulin. You may remember that insulin is a small protein hormone made up of a 21 amino acid A chain and a 30 amino acid B chain, disulfide linked. And the fast-acting insulins are made by replacing the single proline residue that appears at position 28 of the B chain. 2 of the fast-acting insulins on the market are referred to as Aspart. This is also called Novolog. It's sold by Novo Nordisk, and it changes the proline at B28 to an aspartic acid. Lispro or Humalog is sold by Eli Lilly, and what they do is to flip the proline and the lysine at B28 and B29. In my opinion, what they're really doing is just moving the lysine into B28. It's not terribly surprising that messing around with B28 would cause a change in hexamer stability, because if we look at where B28 is with respect to the dimer interface in the hexamer, there are 2 copies of B28 directly adjacent to that interface. And so if you disrupt those intermolecular interactions, you'd expect the hexamer to come apart faster. That works. If we look at insulin activity as a function of time after injection, regular insulin will peak after 3 or 4 hours, and the fast-acting insulins will peak after about 90 minutes. But it comes along with a penalty, and that is that the faster dissociation leads to faster fibrillation. You can do these fibrillation assays in a lot of different ways, and so you can find lots of different numbers for fibrillation times in the literature. But here's an example of conditions where human insulin fibrillates in about 3.5 days. Aspart fibrillates about twice as fast. We had a conjecture here that the reason that this was happening is that when you replace proline at B28 through any conventional mutagenesis, you have to replace it with an acyclic amino acid. Proline's the only cyclic one. And so we know that the conformational flexibility associated with proline is lower than that of any other amino acid. If we look at the Ramachandran plots, which tell us the observed phi and psi backbone dihedral angles, for proline we see occupation of a small fraction of conformational space. For all the other amino acids, we see evidence of much greater conformational flexibility. And so our conjecture was that that fraying of the chain ends is made even worse when you remove the configurational constraints of a proline through the introduction of an acyclic amino acid. Evidence for that comes from the crystal structure. These are the C-terminal regions of the B chain for wild-type insulin, shown in white. This is the proline, and it causes the kink in the chain that's characteristic of proline residues. This is aspart, or Novolog, in that same region, and the chain trajectory is markedly different. And so we think that this disrupts the dimer interface, but it also causes the chains to fibrillate faster. So what do? You want to be able to engineer those interchain interactions, but you don't want to lose the cyclic structure of the proline. And so what we have done over the last couple of years is to explore the introduction of non-canonical proline residues into insulin at B28. All of these work. I'll show you the results in a couple of minutes, but I'd like you to focus on the two that are shown in blue. They turn out to be the interesting ones. They are both hydroxy prolines. They're hydroxylated at the 4 position. This one is the cis form, or the 4S form. This is not found in proteins naturally. This is the trans, or 4R form. This is abundant in collagen, so this in some sense is a natural amino acid. It's just not encoded in the genetic code. It's made by post-translational modification. And what we're going to see is that these two amino acids affect the properties of insulin in strikingly different ways. So the stereochemistry matters a lot, so please pay attention to the stereochemistry. So the first question is, can you make insulins with these prolines in them? They're made easily. We make them in the form of proinsulin, and then we mature the proinsulin into a properly folded form. Under our conditions, we make about 40 milligrams per liter of wild-type insulin. We reduce the yield somewhat when we use the non-canonical amino acids, but the 2 hydroxylated forms express at levels that are close to wild-type. I show here that we get about 90% replacement. That was actually true of some early work. We have now gotten this up to virtually 100%, and as it turns out, the results don't depend on that difference. So we can make them in good yields. The first question is, do they work as insulins? Do they reduce blood glucose in diabetic mice? We collaborated with colleagues at City of Hope to examine this. What we're looking at is blood glucose concentration as a function of time in minutes after injection. And what you can see is that human insulin looks like this, aspart looks like this, and the 4R and 4S hydroxys are essentially identical. Now you might be wondering, why don't I see a difference between fast-acting insulins and wild-type insulin? And the answer is that in mice, you never see such a difference. The subcutaneous tissue structure is different enough in mice that you don't distinguish fast-acting from normal insulin. So this was just to verify insulin activity. We then began to look at the insulin biophysics as a function of substitution. And so the first thing that we wondered about was the monomer-dimer equilibrium that you observe if you remove zinc from the preparation. If there's zinc there, it forms a hexamer. If you remove the zinc, it forms either a dimer or a monomer, depending on the stability of that dimer interface. And so we can probe this either by using analytical ultracentrifugation or by circular dichroism. I think the ultracentrifugation data are easier to understand, and so we're going to focus on those. In this kind of an experiment, what you're measuring is the sedimentation coefficient, which is a surrogate for the molecular size. And the key thing that we're looking at here under these conditions, 60 micromolar insulin, absence of zinc, there are 2 species expected. dimer, which would be over here. The wild type is a dimer. And aspartate is a monomer. So those were our markers for monomeric and dimeric forms. So what we found was that 4S was dimeric and 4R is monomeric. We were very excited about that because we thought that 4R proline, hydroxyproline, was causing dissociation of the dimer, and that characteristic correlates with fast onset of action. So we thought 4R hydroxyinsulin would be a fast-acting insulin. We were wrong. When you actually look at the rates of dissociation of the hexamer, this is what you see. We're looking at the hexamer signal, spectroscopic signal here. We add some terpyridine, it complexes with the zinc, and we read out that spectroscopic signature. The zinc comes out as the hexamer dissociates. So we're looking at that residual hexamer as a function of time in seconds, and the blue and black curves here are wild type and 4R hydroxy. This is the one that we thought would be a fast-acting insulin. It's indistinguishable from wild type. If you look at the relaxation times, they're both about 100 seconds under these conditions. But the interesting thing was that 4S hydroxy dissociates almost as fast as Lispro. Lispro dissociates in about 60 seconds, Lispro in about 50 seconds, 4S hydroxy in about 60 seconds. So this was interesting. It seems to be a different mechanism for speeding up the hexamer dissociation. It's not destabilizing the dimer, apparently, but it is speeding up hexamer dissociation. But the really interesting result came in the fibrillation experiments. These were done by Kat Fang and Seth Lieblich, who've done all of the insulin work that I've told you about so far. The fibrillation assay is a dye-binding assay where you add thioflavin T to a solution of the insulin. You shake it at 37 degrees, and thioflavin T will bind to the fibrillar form of insulin and turn on a fluorescence response. So under these conditions, we're looking at the THT fluorescence as a function of time in hours. You see that both wild type and Lispro fibrillate in about 7 hours. 4R hydroxy seems to fibrillate even faster, but that's okay. We weren't really interested in that molecule anymore anyway, because it's not dissociating any faster. But the exciting result was this one. This is 4S hydroxy down here. In this experiment, it didn't fibrillate at all. Now, this experiment only went out to 18 hours. Seth and Kat didn't expect the stability of 4S hydroxy, and they went home after 18 hours. Can you imagine that? Graduate students going home after 18 hours in the lab. What we found, of course, was when they came in the next day, that they redid the experiment, and in fact, 4S hydroxy insulin does fibrillate out here. But it's about 4 times more stable than wild-type insulin. So it's a fast-acting insulin that not only preserves the stability of wild type, but enhances the stability of wild type. Why is that? Why do we see this kind of behavior? We don't know, but the best hypothesis at this point, meaning the only hypothesis at this point, comes from the high-resolution crystal structures that we have of the hydroxylated and wild-type forms. This is wild type over here. This is the interesting 4S hydroxy here. This is the less interesting 4R hydroxy over here. This is proline at position B28. And the one feature that we can see in this structure that we think might be important is the suggestion of a strong hydrogen bond between this hydroxyl group on hydroxyproline and a backbone carbonyl across the dimer interface. Now, why should that give us the behavior that we observe? This is a simple explanation. It may not be right, but I think it's consistent with what we see. This is the path from the hexamer to the fibril. Let's imagine that that hydrogen bond is stronger in the dimer than it is in the hexamer. That'll pull down the dimer energy. That should speed up hexamer dissociation because we're stabilizing the product, and it should slow monomer formation and fibrillation because we're stabilizing the starting material. I don't know whether that's true or not, but that's the current working hypothesis. Now, I was going to tell you a little bit more than that, but I really don't like lectures that go on too long, including my own. And so I'm going to skip a few slides at the end. If people have questions about them, I'd be glad to talk about them. This is just a summary of what the other non-canonical prolines do in terms of dissociation and fibrillation times. And then we've been able to speed up the fast-acting form of insulin, and we can speed up Lispro to make it faster than it was before by introducing a fluoroproline at position B29. But I will skip those, and I will go to the acknowledgement slide. In particular, everything I really had a chance to tell you about was done by Kat and Seth. The work that I didn't have a chance to tell you about, that I skipped over right at the end, was done by Alex and Stephanie. We've been supported here by Novo Nordisk and by JDRF through a grant from Protomer Technologies. I am one of the co-founders of Protomer Technologies, and Protomer Technologies is trying to develop this form of insulin therapy and other forms as well. So I'm going to leave it there. You've been wonderful hosts. I'm delighted that there are some people here for lecture number 2. That's great. I'd like to thank you for your attention, and I hope there'll be some time for comments and questions. Thanks.
And briefly, before we take questions, we would like to present to our lecturer the award. So David, thank you very much for your lecture and for all of your stewardship in education, chemistry and chemical engineering, and MIT. Thank you very much, David.
Thank you, Paul. Thanks to all of you. Thanks. All right. So do we have time for questions, or do we?
Plenty of time
Yeah?
for questions.
Okay.
We'll open it up, and I think we have microphones on either side. People can just walk up.
Wonderful work, David. I wanted to ask you, as someone outside the field, if we wanted to use these techniques to make a truly synthetic polymer, right? The prospect, we can't build the cell or the organism out of synthetic amino acids, right? So the prospect of depleting every amino acid, or even many of them, and then supplying the synthetic precursor is not possible. But so I wonder if the future direction is maybe to go to cell-free systems or go to multi-phase or solvent systems. I mean, is that fantasy, or could we actually think about making completely synthetic polymers in this way?
Yeah, I don't think it's fantasy. I think cell-free systems are probably a very good line of investigation, line of development in this area. But the situation is a little bit better in cells than one might think, because if you make these kinds of proteins in batch processes, that is, you grow up the cells with lots of the normal amino acids in them, you then separate the cells, deplete the natural one that you don't want in there, put the artificial one in, and then turn on the synthesis of the protein of interest, you can get good yields of protein with the non-canonical amino acids in there. Now, most of those proteins, the sensible ones to start with, have modest numbers of substitutions. We've certainly made periodic polypeptides where we've had multiple sites of substitution. That works pretty well. I think it should be possible over time to increase the content of non-canonical amino acids, but I don't imagine in cells that we're going to be making macromolecules entirely out of non-canonical amino acids. And so I think one of the challenges is to figure out how you get the changes in properties that you need at relatively modest levels of substitution.
Hi, thank you also for that wonderful talk and the wonderful work you've been doing. Had a question about immunogenicity. I'm guessing you're asked that with some frequency. Endogenous insulin creates a negative immune response in a lot of patients. Do you think that with these non-native amino acids, that possibility might increase, or do you have reason to think it will be affected?
Yeah, it is a very good question, an important question that will have to be answered, I think, on a case-by-case basis. The only experience that we have directly with a non-canonical amino acid in a protein therapeutic was developed at another company that I helped to start about a dozen years ago called Allozyme. They made a pegylated interferon beta for treatment of multiple sclerosis. There's now a commercial form of pegylated interferon beta, not made by Allozyme, made by Biogen IDEC, using more conventional pegylation chemistry. But at the time that Allozyme was getting going, they were looking for new pegylation strategies. There wasn't a pegylated interferon beta at that time. What they decided to do was to replace the N-terminal methionine with azidohomoalanine and then click on a polyethylene glycol only at the N-terminus. That worked extremely well. That material stayed in the circulation for 3 weeks. The hope was that it could actually go to monthly dosing with that material. It went through 2 phases of clinical trial, got approval for a phase 3 clinical trial, but the cost at that point was projected to be $150 million, and Allozyme was never able to raise that $150 million. In the meantime, Biogen IDEC was coming along with their version, and I think that probably made it more difficult for Allozyme to raise that money. That's a long warm-up to the observation that in that context, because this material was put into human patients in the first 2 phases of the trial, there was no evidence of an immune response to the interferon beta itself. There actually was a little bit of an immune response to the polyethylene glycol, which is becoming more and more widely expected in these sorts of situations. Now, I think it would be fair to say that that's not a very stringent test, because at least most of the sites were probably pegylated, that most of the non-canonical sites were pegylated, and so the immune system probably would have a hard time finding them. So I don't know that there's much to go on at this stage. My guess is that hydroxyproline, even though it's the non-natural stereoisomer, my guess is it won't cause much of a problem. As you may know, as you incorporate more hydrophilic units into protein chains, immunogenicity typically goes down. Now, I wouldn't want to make that as a blanket statement, but I don't think there's anything about the structure of hydroxyproline that's particularly worrisome in terms of immunogenicity.
Thank you.
Thank you.
So you mentioned mostly focusing on the synthetases in terms of trying to recode. Have you utilized the degeneracy of the code and tried to change some tRNAs such that only specific codons would be changed to the amino acid or whatever that you wanted, given that there's some codons that aren't used very often in E. coli or other organisms?
Yeah, another really good question. We have done some of that. We were able to split the degeneracy of the phenylalanine codons. There are 2 phenylalanine codons, but there's only 1 phenylalanine tRNA. And so that tRNA reads one of the codons through Watson-Crick base pairing, the other through Wobble base pairing. And so what we did was to make another tRNA that was a Watson-Crick base pair match to the second codon. We could get pretty good discrimination between the 2 codons in that experiment, but it was never as high as we would like it to be. I think there are probably good strategies for improving the fidelity of that process. We're not currently working on it, but I think that would be a productive line of investigation. And then, as you may know, the other strategy that's been used to do what you suggest is nonsense suppression, where a suppressor tRNA is brought in to read the stop codon and to deliver the amino acid in response to the stop codon. So that's, I think, another version of what you're suggesting. The 3 codons that are ordinarily degenerate in terms of specifying a stop, you've broken the degeneracy of those 3 codons.
Do we have any further questions? I had a question for the speaker while our audience is thinking. I was going to ask if essentially this problem of immunogenicity with PEG, which is something that those of us who develop drug delivery systems think about all the time, might be addressed by some sort of hybrid polyamino acid, and if this is a question that you've begun to look at or anyone in the field.
Yeah, we have not. but some of you may be aware of Pim Stemmer, one of the people who developed the field of directed evolution, along with my colleague Francis Arnold and others. Pim passed away a few years ago, but one of the very nice things that he did, in addition to his work on directed evolution, was to prepare artificial sequences that were essentially invisible to the immune system. And so one of the reasons for my answer on the question on immunogenicity was the success of that approach. This molecule is called X10, X-T-E-N, and it is essentially an uncharged hydrophilic sequence of amino acids that Pim was imagining could be just encoded into the gene for therapeutic protein, and then naturally make a solubilized, non-immunogenic, long-lasting form of therapeutic proteins. I don't know to what extent that technology is being developed. We've actually used the X10 sequence for other reasons in our own laboratory. It expresses extremely well in bacterial cells, so if anybody can see a use for it, I would recommend it. It's a great sequence.
Any other questions from our audience?
Yes, I'd like to ask what position you played on the baseball team.
Thank you for asking. I played 3 years at third base, and then my coach discovered that I was not a third baseman. And my senior year, I played second base. And as you may have seen on the slide, the 1974 team went to the NCAA tournament. We lost both games we played in the NCAA tournament. If it had been a triple elimination tournament, we could have lost 3.
Definitely a Go Tech man. Any other questions? If not, let's thank our speaker one last time.
Thanks.